All right, we're going to start from this slide. I'll have other videos where we go through all of the slides, probably in full. I went through partially the slides once before. I wanted you to watch the videos I gave you on mapping and crossing over first. This should make this a little easier to understand here when we talk about the mapping cross. So I'm here on slide 11. And we're going to begin talking about mapping. And if you've watched the two videos that I made on mapping and on crossing over, this should make a little bit more sense than if you didn't have those. So please watch those two before you watch this one. All right, so let's take a look at what we're doing here. We're doing mapping crosses A, B, and C, and you'll perform mapping crosses on 3 different marker genes. Now, the marker genes are the ones that we kind of know where they're at. We know they're there, and we're going to kind of be trying to find out where the other genes are in reference to these marker genes. So realize that you have an unknown marker on males. You have a mutant here on males, along with a dominant wild type and a marker recessive mutant plus wild type males on this side. Just keep these in mind right here. That realize these are the flies that you should be like mating, all right, or that you should be holding out if you were doing the crosses. So that means these are the ones that you'll be putting into FlyGene to get your results. So this is a bit more relevant to actual mapping here, this picture, and you'll need to be able to do these eventually, make a little graphic like this. So this is how you kind of keep track of it. So what we have here is wild type, here's a mutation, here's our unknown, and this is a female. So we're going to take this female and we're going to cross it here. And we're going to look to see if, okay, we're going to cross it with this male. And we're going to think, well, what should we be getting? Well, we should only get, normally, without crossing over, you should get chromosomes with your unknown and the wild type, and then one of these two. Well, these two are identical. So you should always get unknown and BW. You should never get wild type over here on this chromosome. So there are certain things that should never happen without crossing over. If you get these things crossed up, like what do you never get on the same chromosome? Here you get unknown and BW. Here you should never get 2 chromosomes. You should never get this again. None of your males should be homozygous for both of these, and your females should never be homozygous unknown, and they should also never be homozygous wild type. That shouldn't happen. So there are some things that should never occur when you cross these 2 flies. But if they do, you count that up, and that's how you're going to find out the distance between your unknown and one of these marker genes, like BW, or depending. So here's the parental, all right. Here's your recombinant, all right, and you get percentages. And this still isn't corrected here, but I think we can do it. Here you're going to collect how many phenotypes you have of a different type, all right, and add them up. And then you're going to add up the ones that you are not expecting. All right, these are the ones in red here, and you should never get. Let's look at this. Unknown BW. Is that right? Should we ever get unknown BW? Well, if I cross this with this one, I should get what? I should get unknown, and this will make it unknown, 2 unknowns, but yeah, you still had a plus, so therefore it's heterozygous, and assuming the plus is dominant, you won't see that phenotype. So unknown and BW, you should never see that. So that makes sense. What's the other one they give you? Plus. All right, so should we ever see that one? Is there any way for us? No, because this one, if you're inheriting one of these, this one comes over here and you combine it with either one of these, you'll get 1+, but you're never going to get two pluses. So you should never see plus plus, and if you do, well, then you know that either this one exchanged here or this one exchanged here, and because of that, then you know that you got recombination. So the idea is you have to figure out, and that's really the hard part, tell you the truth, is figure out what you shouldn't be getting. Or maybe you just run your crosses and figure out what you should get, and if you see anything different, then you add it to this. But really you should also be trying to figure out ahead of time what you should be looking for over here as far as your unknowns. And if you get weird numbers, all right, I shouldn't be getting, or I should say, you get weird crosses, you're getting some mixtures that you don't think you should have, well, that's probably an indication that you have crossing over. And you use those numbers there in red, you add them up, all right, it's your total, but you're going to add up these two numbers, the two in red, and you're going to divide by the total of all of these, and that's going to give you the number of mapping units. So hopefully with the two previous videos I gave you, this actually makes a little bit more sense, at least in why you're doing what you do. And this is showing you the mechanics of how you do it. So just keep in mind, find the flies that you shouldn't be getting under normal circumstances, normal crosses, and you add those numbers up for those types that are odd, all right, that seem like they shouldn't occur. Add those up, all right, divide them by the total number of progeny, and you've got your number of mapping units. So we talked about hypotheses and how you should state those. That was in a previous video. Here's another example. Of course, you take the ones you don't think you should get, you add them up, and you divide by your total. It's always same. So notice this one's a little closer, right? It's a smaller number, it's closer, because remember, when they're closer, there are less opportunities for crossing over. And if they're absolutely necessary, as in survival is absolutely necessary for them to be close to one another, then you won't get them at all. You don't get any crossing over. Okay, so keep that in mind as well. They also could be on a different chromosome, because they also could be very far away, but that's a different issue as well that we'll cover later. So you also can figure out, if you have two markers, you can figure out that where it is between the two markers. So it's always a good idea to do this twice, and then you can say, hey, it's 11 from this one and 12 from this one, and you can kind of estimate and see if they agree. That's a good way to double check your answers. But realize the distance, as I noted in my video, you don't know if it's positive or negative, so you either take the location of your marker and subtract the distance, or you add the distance. And you don't know unless you do this another time with another marker that you know where it is, or if you're near the ends, you also might be able to figure it out. But in other words, if the marker was at the end, you know you can't get too small, or too big. You can't go off the chromosome, so you only positive or negative. But then in the other case is if you get two markers, and then you kind of can find that it's 5 from this place and 6 from this place, that'll kind of tell you which direction you need to go. So usually you do this with 2, maybe 3 markers, and try to pinpoint exactly where your gene is located. You will have to learn to do the math for this, all right, regardless of whether or not we're doing this in the lab or not. All right, so you would do this, run it on FlyGene, and come up with your distances. Now, when you're using your unknown versus 3 markers here. So you did this 3 times. You compared your unknown, you did some crosses, and came up with numbers that have to do with your distance from the dots, purple and brown. And it said you're 65.5 from here, 65.6, and 61.2. And your average is then 64. And that's about as good as you can do. So you figured out where each of these were, and it gave you a location, where it might be. This is actually not a good, this is kind of confusing here, because this is giving you a distance from one place. Yeah, I don't like this. Let's ignore this one, because I don't think this gives you the right idea of how this works. This one might do a little better. um, realize you can figure out which chromosome your gene is on by doing crosses on different markers on different chromosomes. And if you know that if you know which chromosome your marker is on, and you don't get any crossing over, well, there's a good chance, well, your gene is not on that chromosome. I mean, that's actually a very straightforward idea there, but just keep that in mind, that you need to find out what chromosome your marker is on and make sure it's on the same chromosome as as your unknown. Now, you guys are going to have an advantage. You can go look it up. But previous semesters, they didn't have that knowledge, so they had to do lots of these to figure out whether or not it was on the correct chromosome. Oop. All right. To L 0 to 55, un is here, so 55. I don't know what they're trying to tell us here. The size, I guess, the lengths, 0 to 66. 2R, so this is the chromosome that your unknown is on. All right, now eventually we'll get to the nucleotide sequence. And today, really, this is the way you would do this for sure. It's maybe a little harder, but it's getting cheap enough that it makes more sense to go do sequencing than it does to just breed flies. So if you don't have a fly lab and you have a gene and you want to know where it is today, they just send it off and to a lab, and they sequence it and tell you pretty much where it's at. In the past, you had to breed flies like we're doing. And if you have a fly lab, it's easy to do. But if you want to know it tomorrow, then you would just spend the money and get it sequenced. And that's why we learn bioinformatics today. It's the trend. Slowly, we're not doing things this way anymore. We're doing them with the sequencing. All right, now a lot of you have asked me about how to use FlyGene. This slide gives you an idea. And now I made a video showing you this. Please watch that one in addition to this, but I'm going to go over it again, and I'm going to put these slides up so that you have access to them on the OneDrive. Realize the genes are here. All right, so you type in the name of your gene. Oops. Right here on Flybase, all right, and you should get genes here, and there are lots of things you can do. You can go look at the transcript, the alleles that are involved. There are many things to look at. You can right click here and find the chromosome arm that's on. That'll give you the actual location of your gene. It'll give the arm, and essentially it's 2L, and then you can find the actual location here with one more click. I'll leave that to you, but actually here's the sequence location and the arm location. You have pretty much everything here you need on Flybase to find the location of your gene. I'm going to leave it to you, give you a little bit of work to do on your own, but when I tell you the sequence location, look, it's on 2R, and here is the exact sequence, the exact location and numbers, 21,559,977 to 420 here. Let's see, anything else? The genomic location. Remember, when you're doing this, you go down here and click on these below. So you scroll down, and there are lots of things down here that give you lots of information, pictures. Everybody's asking me about pictures. Where can I go? A lot of them are down here. You go search through here, maybe phenotypes, things like that. You should be able to find some pictures. You can look for specific alleles. All right, now the 1s and 2s depends. If you're having a hard time finding alternatives, once you know, say your fly is a different color, well, you need to go find flies with the color is the difference. And I put out a video and some explanations on how to go do that. I gave you one very good video. I think that that I did not make, but I gave you a link to it. You should use that one. Please watch all the videos I tell you to watch. So here, click Convert to Genes. Pretty straightforward if you're looking at it. Chromosome arm again. And cytology, you don't really need to know this yet. Cell types and things like that, where it's located. But these are things that are phenotypic. In other words, what cell types do they do? What that's what their cytology and things like that are. So you can, of course, map position contained in region right here. So you're getting more information. Realize here is there's just more information for you to include in your paper. That's what you're really doing here, is you're collecting information for your paper. So make sure you read through these. You can read here. You don't actually have to read through the PowerPoints. You can just watch this, and if this is visible to you and readable, then you've got it. Again, more phenotypic description, all right? Tells you more about it, and it tells you the paper to go find. When you look it up, you can use things like Google Scholar, etc. And you need to learn to search for these things. The NCBI PubMed is another place. Maybe I can give you a video on PubMed. Much more information here. You have to go in here. A lot of you, I've been pointing you to places like this when you ask. But I really expect you to find this more or less on your own. Now eventually we're going to do more chi-squared. I've already put out videos on chi-squared. I went from the evolutionary perspective. But what we're going to be doing next is we're going to find out if the number of progeny you have are correct. In other words, are you getting the right numbers? Are they significantly different from what you should be expecting? Like you're supposed to get, you do a Punnett square and you're supposed to get twice as many blue flies as you do green flies, and well, the numbers aren't quite twice the number like you thought you were going to get. They're close, but not exactly. How far off can you be? Well, you use chi-square again to check that, and here's just an example of that. So go through this slide and understand how to do the chi-square for it. This would be the exact same thing here. So remember, you associate this with a hypothesis, and depending on your number down here that you get, ultimately you use your chi-square value in the table, and if you're above or below it, you either accept or reject your null hypothesis. This is stating it exactly correct here. All right, and these are a little off here. The print on the PowerPoint here, but you can still read it, I believe. All right. So overall, now here we do a null hypothesis, and we were doing this earlier. This is why I didn't like this one, but I see now that what they were doing is this number was outside your range that you received from acceptable range that you received from your chi-squared, so you have to eliminate this one. So you redo it and redo your average now without the dots, okay? And you just use those two because that just wasn't a correct number. That was too far outside the acceptable boundary for you to use it in your research. Okay, if you have more questions on this, I'll be doing more chi-square analysis for you, and I've already given you a couple of videos involving chi-squared, so make sure if you don't remember how to do them, you go back and review those and then come back to these PowerPoints. Now here is our mapping again, and this is of course the interval. Once you've decided where you're at 65.5, you need to make a little graphic like this. Now maybe not with WA1, but eventually you're going to have to. And so we believe that it's at 66.7. We had some mapping genes that told us where BW was, and SCA, and PR, and B. So we know where these are. All right, these are known. And our unknown, maybe we mapped this to each one of these. We found out that, oh, when I cross my unknown flies with the bee flies, I find that I get a certain number of flies that do crossing over, and it says that they're way over here. So I should be getting, when I cross unknown with bee, it should be indicating that I'm getting a lot of flies with a combination that normally shouldn't be there. I get a little less, I get fewer flies, I guess you would say, when I do it this way. I get actually a whole lot when I go with BW. All right, so there should be, when I cross unknown with BW, I should be getting lots of these strange flies that I wouldn't normally get under a regular Punnett square. But when I cross it with SCA, I am very close. I should get almost none, maybe none. Here it looks like you got maybe 1-2% or something. Very few flies, so therefore that means you are very close to SCA. And then you just kind of figure out where you are between all four of these, sorry, and you can get a very precise location as to where your unknown is located on chromosome 2. This is assuming that you've got access to 1, 2, 3, 4 marker genes on FlyGene. So you have chromosome 2, you find these 4, you find out where they're located, you cross flies with your unknown, and wherever you, however many crossing over events you have should show up in the number of flies, and you use that to calculate your distance from each of these, and then you take that and figure out exactly where you are. You'll find out that, oh, I'm so far away from PR, I'm a little further away from B, and I'm a certain amount from BW, and I'm right next to SCA. I should get very few recombination events going on with SCA when I do this cross. All right, so hopefully this explains it a little better for you. I'll have more videos, and I'll put these PowerPoint slides up on the OneDrive.